Life as we know it is beyond fascinating. So many beautiful feats that appear simple, yet are complex, that appear naked to the human eye. Thousands upon thousands of biological and molecular actions taking place consistently to allow you to do anything from tying your shoe to performing an exam, and so on. Cells are the reasons you are who you are and how you are you. Cells carry out complex mechanisms to keep the body functioning at exceptional capacity without any interruptions. Cells are the functional units of the body. Cells are incredibly small, ranging from 10 micrometers to 100 micrometers. There are many components to a cell that assist in carrying out specific tasks, such as the mitochondria providing much of its needed energy to live. One component, the nucleus, houses our DNA, a genomic sequence that codes our cells. Each person has a unique set of DNA that defines themselves from others, such as certain genes containing a different version that allow for the blue color eye instead of a brown. A gene is a sequence of genetic information that is read by our cells to produce various effects that make us us. Multiple genes exist on a singular chromosome, and normally the human body contains 46 chromosomes, 23 pairs, with each coming from a mother and a father, that serve as a foundation for locating, housing certain genes. Simply like a command center of the cell, the nucleus contains DNA to determine our identity and dictate our daily function. The functional units of DNA is referred to as genes, which encode proteins that are the main players to execute cell functions. The DNA in the command center is heavily regulated information that is usable to create and sustain various functions of said command center. There are different types of cells that carry out larger functions of the body, such as neurons that allow your body to send signals for muscle contraction so you can perform movement, like picking up a pen. The difference is certain chromosomes in your genomes are silenced, and some are expressed to prevent a cell from performing poorly or not performing the right action at all. This differentiation is what allows your body to do the things they need to do. Sometimes diseases may arise that inhibit the normal functions of the body. Typically, cells replicate at an incredible rate to replace old, dying cells that cannot perform their original functions. Diseases, though, can damage the body in a chaotic way, prevent these cells from keeping up, as well as fixing disease itself. For example, a German boy with epidermolysis bullosa, which is causing fragile, blistering skin, lost 2/3 of his skin to a genetic mutation within the LAM3 gene. Every treatment seemed ineffective, even a skin transplantation. As a last resort, Dr. Michele De Luca, an Italian stem cell expert who heads for the Center of Regenerative Medicine at the University of Modena, performed a treatment that involved collecting the remaining skin from the boy, correcting the genetic mutation from the skin cells, and then culturing the skin into sheets for transplantation. He was able to make a full recovery after 21 months thanks to these cells, also known as stem cells. Stem cells are a small amount of cells in your body that are unspecialized cells that can make copies of itself and produce multiple types of more specialized cells with particular functions. These two characteristics are what define what stem cells are. Stem cells can be committed to becoming specific cell lineages in the tissue, or completely uncommitted as possible to allow for proper development to a particular type of cell. Tissue stem cells are an example of this, and they help regenerate and replenish designated lineages in adult tissues. Some examples of tissue stem cells are hair follicles, skin, intestinal epithelial cells, also known as the gut lining, and hematopoietic cells, which are originating in your blood and immune cells. Here, an illustration shows multiple locations of where small amounts of stem cells reside in areas of your skin to allow your skin to regenerate new tissue every day. The gut stem cell is another example of how a multipotent stem cell may become progenitors, also known as intermediate cells. to serve as different functions to make the small intestine functional when absorbing nutrients from the food that you digest. A significant clinical application for certain treatments is the use of hematopoietic stem cells. These cells reside in the bone marrow and can branch into two different areas of functionality, myeloid and lymphoid. The myeloid is responsible for more of your red blood cells, platelets, and certain immune cells like basophils. Lymphoid cells can become T cells or B cells that help with humoral immunity, such as responsibility for remembering a pathogen to fight off your infections or sickness, also known as antibodies. There are many fascinating applications for the utilization of these cells to treat incredibly challenging diseases such as aplastic anemia. Understanding how stem cells balance self-renewal and differentiation is critical. Most stem cells are somatic cells, while one type is the only germline stem cell in the adult tissues. These are known as spermatogonial cells. They allow for the replenishment of sperm in the testes. They exhibit a very methodical mechanism as to how this balance is achieved. Mentioned earlier, another clinical application is hematopoietic stem cells. They originate in the bone marrow and are responsible for providing a self-sustaining supply to blood cells of all types. This involves autologous, or same person, or allogenic, which is the donor, transplantation. The stem cells are collected from the patient, and then the gene responsible for the disease is edited in cell culture, and then finally transplanted back. Dr. Cunningham from the University of Chicago, chief pediatric oncologist, helps explain how different types of transplantations can help patients with certain diseases.
The challenges with autologous cells that, when you suck the cells out of the bone marrow or mobilize them, they may have some tumor cells. And so it's usually the autologous cells are usually for a very restricted group of patients. So we know in neuroblastoma that although there may be some residual tumor cells in the cells we infuse back into the patient, those cells probably die relatively quickly. And so we're not just reseeding tumors into the patients. But in other diseases, we have found that that's not the case. And so we don't use autologous transplants for very many diseases anymore. The patients need to be in remission. In other words, there's no obvious sign of disease. So again, it's a very limited group of diseases we use autologous transplant for. Allogeneic stem cells, which are obtained from either a sibling or a matched unrelated donor, or perhaps from a parent, that's called a haploidentical donor. Those are used for leukemia, for lymphoma, and are also used for some genetic diseases of childhood, such as Diamond-Blackfan syndrome, which is a deficiency of red blood cells, or they're used for aplastic anemia, which is where the bone marrow just stops working completely. So there's a lot of different uses.
The process behind stabilizing hematopoietic stem cells has been researched on for some time. A significant challenge is obtaining a sufficient amount of hematopoietic stem cells. With a fixed amount from a donor or patient themselves, we would need significant amounts of hematopoietic stem cells from other sources. A solution to this is expansion of these cells before transplantation. Scientists optimize conditions to support tissue stem cell self-renewal and have made some progress in certain tissue stem cells. For example, we would need to add cytokines to support the self-renewal of stem cells such as hematopoietic stem cells, including SCF, TPO, IL-11, FLT3L, and IL-6. But in general, tissue stem cells are hard to maintain long-term in vitro, also known as outside the body, with unchanged to their stemness and differentiation potential. The amount of treatments are seemingly endless. Stem cells with advanced gene editing techniques enable treatment of previously untreatable disease, such as AIDS. Hematopoietic stem cells, mentioned earlier, for producing T cells, can be genetically corrected to inhibit the production of a surface marker called CCR5 that HIV uses to enter the T cells and utilize them for their own proliferation. At Cornell Medicine, a stem cell transplant specialist team and an infectious disease specialist led the clinical trial for this accomplishment recently, with the results that include HIV remission for a second time since 2009.
We actually have a clinical trial here, which is a national trial for CRISPR gene therapy for a sickle cell disease. We harvest stem cells. We purify those CD34 cells using a column that actually allows us to purify the stem cells from all the more mature cells. And then we incubate those cells with a cocktail of molecular biology agents that actually are the nucleotides that actually target the specific site in the genome. And then we do that editing. We test to make sure that the editing has occurred. We freeze the cells, and then we prepare the patient for transplant using chemotherapy, and then we actually transplant the cells back into the patient. One would hope, as someone who actually has taken care of children with blood diseases for many years, that in the next 20 years or so, most of those genetic diseases we can actually correct with this kind of approach. But we're at a very early stage in the clinical testing of that strategy. So we don't know for sure how many patients and how, we probably will see complications we haven't thought about yet. I'm confident they shouldn't be any that will be very challenging, but you never know.
All this considered, adult tissue stem cells are nature's miracle, almost seemingly immortality factor. However, there are significant challenges that still must be overcome to achieve adult tissue stem cells from being utilized to its maximum potential. For instance, there are some tissues that do not have stem cells to replenish dead cells, and it is thought for good reason, such as heart cells. Next, some stem cells seem to be difficult to access, such as the brain. Finally, they are not fully unspecialized. They cannot become any cell in the body, but rather any cell within its designated area of special function. That is where pluripotent stem cells come in. One type of pluripotent stem cell is through embryonic stem cells. When sperm and egg fuse, they produce a zygote that eventually develops into something known as a blastocyst around 5 to 6 days post-conception. Inside the blastocyst lies an inner cell mass that are essentially the pluripotent stem cells. They could be extracted and placed on a feeder cell to nourish them outside the membrane to be kept fully healthy and functional. Simply, two distinct and significant types of stem cells, as previously mentioned, are tissue stem cells and pluripotent stem cells. Tissue stem cells are limited in self-renewal when cultured, committed to certain lineages, and to organs that do not have these stem cells, they cannot be regenerated. They can be found in certain areas of the body, like bone marrow. Pluripotent stem cells are unlimited for self-renewal and can become all lineages in the body, but there are no known pluripotent stem cells that are harvestable in the human body when developed. Today, there are many diseases in which medicine continues to fight so hard to cure. With many interested in regenerative medicine, so much research and findings have contributed to the future use of stem cells that can help improve treatments of diabetes, Parkinson's, traumatic spinal cord injuries, and so much more.